Thank you very much. I'm really happy to be talking at such a diverse and interesting seminar. I'm going to be talking to you today about how some very strange animals see the world in very strange ways, and about what there actually is in the natural world to be seen. I'm a vision science researcher at the University of Lund in Sweden. I really wish I could be in Prague in person to give this presentation. It's a lovely city, but oh well. Anyway, let's get into it. I'm very interested in how animals see the world and how their eyes evolve to see the world. And I'm especially interested in strange animals, animals that have lifestyles and habitats and visual systems that are very different than our own. I'm also very interested in marine biology. Since I was really young, I always wanted to be a marine biologist, and it's been a really lucky thing in my life that I've ended up being able to work in tropical marine environments on animals. And this has also very much driven my research direction, trying to find the best possible ways to do research in coral reef habitats and spend a lot of time underwater. I'm also very much interested in photography. I think it goes quite synergistically with being interested in vision science, because a camera works almost exactly like an eye. And there's a really fulfilling aspect of it where you can try to capture something about the animals you're working on or about the way they see the world with a camera. So the vision science research and the photography goes together quite a bit for me. The reason I'm interested in vision to start with is it's how emphatic and how exciting it is to life on Earth. If you go back before the Cambrian explosion 500 million years ago, this is basically a scene of what life in the oceans looked like. There was a lot of things that are kind of like jellyfish and corals and sea pens that would mostly just be filter feeding through the water or sifting through the soil. So not too excited, kind of a lazy existence in some ways, but peaceful. But then after the Cambrian explosion, things changed quite a bit, and a tremendous amount of drama gets introduced into the natural world. Suddenly there's predators, there are animals fleeing from predators, there are animals that are navigating around their habitats to find more opportune places to feed or hide, or to migrate to different habitats. And one of the major things that is influencing this huge expansion of animal forms and this huge expansion of animal behavior is the evolution of eyes. Once you can see the world around you, once you can interact with things far away at literally the speed of light, you can become a predator and chase things down. You can see possible predators and run away. So vision, among some other important sensory innovations, are probably one of the major drivers of the Cambrian explosion, where all these forms of animals diversified into a lot of the phylum that we are accustomed to today in nature, and this huge diversity and beauty of life. So that's one of the things that really attracts me to vision. To sort of ground you on some of the terminology I'm going to be talking about today, there's basically two ways nature has come up with to make an eye. There is a simple or camera type eye, and this is like our eyes. It has a single lens that passes light onto an array of photoreceptors at the back of the eye, and we integrate information from those photoreceptors in the retina into our perception of the visual world. A lot of animals also have something called a compound eye, where each individual photoreceptor has its own lens, and you stack these all together and then combine information from them to get a vision of the world. And this is very common in things like insects, and I guess is the most common example that people would know, but many other things have compound eyes as well. And looking around nature, there's a tremendous diversity based on these two basic designs. So like I said, we have vertebrates like us have a simple camera type eye, but also jellyfish have eyes that are with a single lens and a retina. So do many mollusks, including octopus, squid, some other cephalopods, and also some scallops. Some arthropods have come up with these types of eyes, like spiders, and also some annelid worms, some pelagic worms have very nice camera type eyes. And on the other side, there's also these compound eyes that you see in flatworms, even starfish on the tips of their arms have compound eyes. Some mollusks have compound eyes along the mantle of their shells. Same with, and then as I mentioned earlier, arthropods like insects have very, very elaborate, nice compound eyes. And all of these evolved independently, and there's a lot of convergent evolution coming into them. A lot of interesting themes of animals coming up with the best way to see the world possible. Another important thing to think about when you're wondering about how an animal sees the world is what light actually is. Light is a manifestation of the electromagnetic spectrum. These are particle wave phenomena that propagate through the universe at the speed of light. And this goes from everything like gamma rays and X-rays all the way up through long wave stuff like radio and TV waves. And light, visible light, occupies this thin sliver of the electromagnetic spectrum. Around 300 to 700 nanometers in wavelength, and 300 to 400 is UV light that we can't see, and 400 to 700 goes from the violet to the red, and that's the light that we can see. And the different wavelengths in nanometers are interpreted by our visual system as different colors. So shorter wavelengths are violets and blues, longer wavelengths are red. When you're experiencing visual information, there's a couple different things to think about. The first thing is the intensity of light. How many of these photons are you catching? A very simple visual system can just tell if light is on or off. Time is also important. If you can compare light intensity over a gradation of time, you can do things like set a circadian rhythm. A lot of simple animals do this. We do this with different photoreceptors than the ones in our eyes. And then once you also get spatial information included, so changes in intensity over time and being able to see more than one pixel of space in the world, you can get what I would consider to be true vision. So this is how we think of vision. We think of a scene made up of pixels or some resolution, and changes in that scene over time as we move our head around or as things change in the scene. And there's other levels of visual experience that can then be plastered on top of this. So for instance, color. We do all of these components of vision and also achieve color information from this. We do color vision pretty well, but a lot of animals can see things that we can't see in color space. The same as some animals can see temporal changes better than us. They can see faster movements. Some insects have temporal resolution that is many hundreds or many dozens of times faster than our own. And also, as far as intensity goes, some animals can see dim light much better than us. And then even beyond all of this, there's another layer that we have zero conception of, and that's polarization of light. As photons propagate through space, they can be oscillating in planes of different angles or literally corkscrewing through the electromagnetic space. We can't perceive this really well, but a lot of animals actually can. Most insects and a lot of invertebrates can perceive the polarization of light, and they often use it kind of like a different flavor of color. So in addition to color vision, they have a whole separate arsenal of polarization sensitivity to tell more details about the world. You can tell a lot about the orientation of the celestial cues from polarized light, and you can also see more details of things in nature from polarized light. So there's a lot of levels of sensory experience that we can see, but there's even more that we don't see that animals are able to perceive. And today I wanted to give you a slight overview of some of the research I do using three examples, three vignettes of my research. I'm going to mostly talk about my work on manta shrimps and how they see the world in a very unique light. And then I'm also going to do some quick stories about fan worms, which actually look at the world with dozens or hundreds of eyes, and also thinking about measuring environmental light fields, actually finding out what there is in the environment for animals to see. So starting up with probably my favorite animal in the world, the manta shrimp. These are really beautiful creatures. They are crustaceans. They vary in size from one species to another, from the size of a fingernail to 40 centimeters. There's 500 described species of these animals. They're incredibly colorful and diverse. Sometimes there's a lot of color differences within a species where the males and females will be different colored, or the colors will be along different lines than that even. And between species, there's a ton of diversity and coloration. I think they're really, really beautiful animals. They live in marine habitats all over the world, but The greatest species diversity of them is found in coral reefs. They're quite exciting animals to study because they're these very aggressive, powerful predators. They have these arms called raptorial appendages that are modified mouth parts that they're able to use as weapons for hunting and for competing with one another. This is one of the most powerful per mass structures and biomechanical structures in nature. They're able to deploy this thing at incredibly fast speeds to attack things. And there's a bit of diversity in these raptorial appendages from one type of Manta shrimp to another. There's a lot of Manta shrimps that we would call smashers that have a big club on their elbow that they hit things with. And then there's also spearers that have these impaling tines that they use to stab soft-bodied prey. And here's super slow motion videos of these being used. This is a spear catching a fish out of the water column, a fish head out of the water column. And this is slowed down many, many, many times. This is an ambush predator, these spearers. Then they jump out of their burrows and snag things out of the water column. And this is an example of a smasher. This is also very much slowed down video. The smashing Manta shrimp here is grappling with a crab. The crab is about to have a bad day when it deploys the club here. And it literally blows the crab's arm off. And these guys are a little bit charismatic in how violent and aggressive they are. Is that after the video stopped shooting for this slow motion capture, this individual picked up the crab's arm and ate it in front of the crab. So they're kind of exciting in that sort of way. But this is one of the most powerful biomechanical movements in nature. And the fact that they can do it in water at these incredible speeds is really quite amazing. They can chisel through reef rock and break out of aquariums with these clubs. They're also really exciting animals because for a crustacean, they have an incredible amount of social interactions that they do. They have elaborate courtship dances that they do for one another when they're trying to attack a mate. They have aggressive displays like this mural spread that I'm showing here, where it's basically them showing off their guns, saying, come on, let's fight. I'm ready. But they've also come up with ritualized combat so that if instead of smashing each other in the face and badly damaging one another, they've come up with this ritual where one animal will hold its shielded tail up in front of it, and the other animal will hit it as hard as it can, and then they'll switch around and gauge the other animal's strike, and they'll decide who's the strongest without killing each other that way. And they use that to resolve territorial disputes. So they're incredibly interesting crustaceans. But the most interesting thing about them is that they have absolutely amazing eyes. So you can see this individual looking around here. You can see both eyes rotate on 3 different axes independently. So they look at different places at the same time. There's different components of the eye that they can use. They keep their eyes very clean. They have special brushes that are just there to clean off the eyes. So vision is very important for these animals, and they've done some really amazing things in the evolution of their eyes to support their life, their really exciting lifestyle. And now just to ground you with a comparison to how our visual system mostly works. This is a simple spectral diagram here with wavelength along the x-axis, which is just the representation that we interpret as color. We have 3 color receptors, a blue, a green, and a red cone, and we also have a rod that we use for low light vision. So we have 4 photoreceptors in our retina, 3 for color vision. This is what the Manta shrimp's color vision arsenal looks like. They have at least 12 color channels, but also have spatial photoreceptors and polarization photoreceptors that exist completely outside of our comprehension of light. You can see they see far down into the ultraviolet below 400 nanometers, and they can also see out into the near infrared past 700 nanometers. So that their experience of color space is much broader than ours, and they have much finer gradations within their interpretation of color space. To just give you a little bit of a rundown of where these different photoreceptors are in the eye and how all these systems overlap one another. This is a Manta Shrimp Eye. They're kind of like other crustacean eyes, except for having this really distinct midband down the center of the eye, which is where a lot of the specialized photoreceptors are. If we look at a cross section of the 2 hemispheres, the areas above and below the midband, These are pretty typical apposition crustacean compound eyes where light passes through a cornea and a crystalline cone lens element and then gets funneled down into these photoreceptors. And in the peripheral regions of the eye, they use these for spatial vision, having a UV receptor and a green receptor, simple dichromatic spatial vision. But then in the midband, they've diversified these photoreceptors into a lot of different roles. And this is where you see the huge expansion of spectral types of photoreceptors for their color vision. And also in the midband, they have polarization vision sensors. They can see linear UV polarized light, and they can discriminate green linear polarized light, but they can also, they're the only animal in the world that can discriminate circularly polarized light that's either propagating in a counterclockwise or clockwise fashion through space. And they've come up with some really, really interesting optics that allow them to make these discriminations. The broader point here is that they are bringing a lot of info into their brains about the natural world around them. So in this staining on the left here is a manta shrimp's brain in the eyes. And you can see that the central brain is actually quite small compared to the optic lobes that are right next to the eyes. The red staining is neural synapse tissue. And you can see they've dedicated a tremendous amount of neural resources to processing the visual information that's coming in through these eyes. And they're bringing together these motion experiences of scanning their eyes around, sweeping this midband that has all the interesting color and polarization analysis equipment in it across the scene to basically color in the world in these scanning eye movements. And they're integrating this all together somehow. And there's still many, many mysteries of how they're actually managing all this information and extrapolating details about their world from it. It's really still an open question of why manta shrimps have built a color vision system like this. No other animal in nature does this with this huge elaboration of color photoreceptors in their eyes. Most crustaceans are simple dichromats with a short wavelength and a longer wavelength receptor, and they seem to exist pretty fine with just that. And even some manta shrimps have scaled down to having a single photoreceptor. They become monochromats, but these species live in very deep or very murky water where there isn't color information in nature. So this has been a loss of color vision capabilities in order to, because it simply isn't needed in the habitat they live in. But the question is, why would they do this? And to bring it back to one of our hypotheses on why the manta shrimp visual system is the way it is, humans have, despite having only 3 color photoreceptors, we see color quite well within the spectral range that we can interpret. We can't see into the UV, which is unfortunate, but within the human visible range, we can see very fine differences in the wavelength of light. And we're able to do that by comparing relative stimulation of our 3 different photoreceptors at very, very precise levels. And we use quite a bit of brain power to do that. Manta shrimp, on the other hand, it seems that they've gone a different route where they're trying to rely less on neural resources to do processing downstream. And they're just trying to catch sort of spectral barcode snapshots of what color something is without having to process the information and looking at fine gradations in photoreceptor stimulation. So they're sort of doing a quick spectrophotometer scan of what something looks like. And one of our hypothesis is that they've come up with this ridiculously elaborate color vision system. It's a different kind of color vision. It's more like a quick snapshot or a scan of things. And we've done a lot of modeling that I won't get into today, but trying to look at how these different color vision and polarization vision systems allow manta shrimps to see one another and tell things about one another. And there does seem to be quite a lot of evidence that they've evolved these eyes to assess one another. There are specific color and polarization patterns that might tell an animal if it's another animal is a spear, a smasher, if it's from the same species as it, what gender it is, if it's a possible mate, and how vital it is. More vital manta shrimps are able to make richer pigmentation and brighter colors on their bodies. So we're still working on this quite a bit, and they definitely are experiencing the light world in a very, very strange way compared to us. And we're still having a really lot of trouble wrapping our minds around how they're perceiving the world. And that's a very much an ongoing area of research for us. And our idea about why this happened is that 350 million years ago, when Manta shrimps broke off from other crustaceans, they already had these raptorial appendages, these huge weapons. And once you have a weapon like that, you need a way to assess one another and to communicate visually before coming to into conflict over territories or over mates. So it seemed like there was a strong pressure early in their evolution to come up with a way to visually assess one another and decide whether to get away or to fight, and instead of killing each other with these giant clubs. So that was a bit about the Manta shrimp work I do, and this is going to now change tack quite a bit to a different kind of marine creature. These are fan worms. And as far as worms go, I think they're essentially the most beautiful worms in nature, and it's about as pretty as a worm can get. They're like these wonderful, beautiful flowers in the ocean. They are polychaetes. They're related to earthworms. They live in these tubes that they build out of mucus and sediment in the substrate. And they project this crown of feeding tentacles up out of their tube into the water column, and they catch little food particles in this crown, and then they pass it down to their mouth. And they also use this crown for respiration. So these are gills and feeding tentacles kind of rolled into one. And because of this, though, this lifestyle of filter feeding, most of the animal never leaves the tube. The only thing that ever comes out of the tube is this crown of tentacles. The animal's head, body, and abdomen are all down inside the tube all day. So any eyes that were on the head, and many polychaetes do have eyes on their head, are pretty useless for this animal, and they've reduced and lost the function of the eyes that are on their head. But instead, they've evolved eyes actually on the feeding tentacles, so they can use them sort of as periscopes to see possible threats coming from the outside world. And these eyes are from a completely different evolutionary track than anything else. They're on a piece of tissue that is completely different than other animals that put a different kind of tissue than other animals put eyes on. And the molecular components and the optical system of these eyes are completely independently evolved. So they're really interesting to study eye evolution on. And also they're nice because they only have one simple behavior, which you'll see in a second. A shadow goes over the animal and it jumps inside its tube at incredibly rapid speed. And that's their one visual behavior. So when we're studying how these eyes function and how they work, it's really easy to zero in on the pressure that evolution is exerting on the system to make a very fast shadow detector or a looming threat detector. And you'll see it one more time here in a moment. This time he takes 2 shadows before he's scared and jumps down into the tube at incredible speeds. These eyes are really interesting to me because in extant species of fan worms, so species of fan worms that are alive today, you can see a whole progression from having no eyes on these tentacles to simple scattered individual photoreceptors to clusters of photoreceptors and on to really nice compound eyes that actually rival those of some insects in their sophistication. And just being able to go into a group of animals that you can find all these different stages on and test what their visual acuity is and how their eye is being used to support this behavior. You can learn a lot about how eyes are evolving in this group. And the other really interesting thing about fan worms is that they have distributed eyes in many species. By that I mean more than 2 primary eyes attached to your head or very close to the brain, like most animals do. There are other animals that have distributed visual systems. For instance, jellyfish, cnidarians, have eyes scattered around their bell. Many mollusks have hundreds or dozens of eyes, like this scallop here, that have these pretty blue eyes that actually have a tapedal reflector in the back of the eye that shines light back at you. Starfish and other echinoderms have eyes at the tips of their tentacles. And then fan worms, many annelid fan worms have dozens or hundreds of eyes scattered all over these tentacles. And it's really an interesting idea to think about how an animal with a distributed visual system sees the world, having dozens or hundreds of eyes overlapping with one another, often on limbs or appendages that are moving around randomly in relation to one another. It seems like a very complicated neural problem to sort out actual useful visual information. it's really hard to imagine how a many-eyed creature would see the world, and to think about what kind of behaviors they can actually support with a multi-eyed visual system. But Manta shrimp, fanworms are really great for this in that some species have gone the other way. They've made consolidated eyes a bit more like ours, where they have a single pair of very large consolidated eyes on a pair of only 2 tentacles, whereas other species have hundreds of eyes scattered all over every tentacle. And both of these systems are driving the same exact behavior, this escape response down into their tube. So it's really interesting to look at these 2 different strategies for seeing the world and trying to figure out how these animals that have very similar brains are teasing out useful information from the natural world using these completely different visual strategies to see the world. And now the last part of the talk I was going to give today is going to talk a little bit about what we actually know about natural light environments. A lot of when a scientist wants to measure light in nature, there's a couple different ways we can do that. We can have a luminance or lux meter that is able to sort of capture all of the light hitting a surface in an environment. And that tells you what the overall light levels are, but it doesn't tell you about where the light's coming from or what some of the more fine spectral and different patterns of light are present. The other way is to take a spectrometer, and that allows us to point it at only specific things. That has a limited acceptance angle of light. And so you can point this thing around and say that leaf is green, sky is blue, the dirt is brown. But these are very kind of crude and limited ways of actually capturing the nature of the whole light environment and animals living in. And so my colleagues have come up with this measurement standard called the Environment Light Field Measurement, and abbreviated as ELF. And this uses a special camera, a commercial, actually a normal camera, a commercial SLR camera with a special 180-degree fisheye lens. So every image you can take with this camera captures half the world, essentially, 180 degrees in one direction. And from the pixels on the camera sensor, you get 36 million radiance readings per exposure. So that's actually bringing in a lot of information about the light environment in the world once this camera system is properly calibrated. And we've come up with a methodology for sampling a habitat where we walk randomly around a habitat, level the camera at the horizon, and take snapshots at a certain time of day or a certain season. And we walk around and we capture all this information, we map it into a flat square of an area, and then we average all of these individual shots together, and we get one of these average scene images that we see here in panel D, where it's kind of blurred together but always leveled on the horizon. And this can give us a tremendous information about the vertical light gradient from straight up above us to the horizon and straight to the ground in each habitat we sample like this. And we get these plots that basically tell us the vertical light gradient. So what the light levels and color levels and the relative color contrast and the relative intensity ranges are throughout this entire vertical light gradient from straight up to straight down. And you can do this in all different habitats and forests and beaches and deserts and not even outside of habitats in human-made environments like the airport terminal on the bottom here. We can also do this at different times of day to see how light levels and relative spectral composition of light changes throughout the daytime and into night, and on nights without the moon when it's just starlight. We can also sample the same habitat at different times of year. This is the same forest in Skåne here in Sweden. Each month of the year where one of these data sets was taken, and you can see how the light environment changes throughout the year from winter to summer and then back to fall. And the composition of the vertical light gradient changes quite a bit. Me being a marine biologist wanted to take this technology underwater and see what the underwater world had to say about its light composition. So we modified a housing to carry this camera and did a lot of diving around the world, looking at marine habitats and some non-marine aquatic habitats as well. And we found there's a lot of really interesting things about the interface of air and water and how that interface affects the light environment underwater. For instance, there is the refraction of light on the surface of water creates this phenomenon called Snell's window, where if you're looking out of the water from within the water, you can actually see a wider area outside of the water than the cone you're looking out through. So you can see things that are far away on the horizon through a hole in the surface. And this is an example of Snell's window here. You can see this very nice window in the surface of the water, and you can see the boat over the edge there, even though it's further away than it looks like in the air. In the water, there's also important phenomena related to attenuation of light. As light goes deeper into the water, you lose short and long wavelengths of light. So at shallow depths, the coral reef might look very colorful and diverse, but then if you go a little deeper and you take some photographs and you bring those photographs up and look at them, you'll see this very blue, green, washed out image that even when you were down there, your eyes adapted to seeing as being colorful, but the actual light that's down there is much more spectrally limited. And there's also phenomenons of internal reflection on the underside of the water surface outside of Snell's window. But to an animal, this could be useful. You might be able to see something over an object because it's reflected up from the surface of the water, then back down to your eye. So there's a lot of things that happen in the water that don't happen on land that are really interesting. And these are some of the examples of the data we collected using this method in the water all over the world. And you can see that Snell's window is in the top of all of these images. It's a very common feature in water until you get to greater depths. And you can see internal reflection sometimes and these other phenomena. Depending on where you are and what is in the water, there's a tremendous amount of variation in color. And this is actually all marine habitats. If we get into some lakes, like in Finland, these tannic lakes, or in the Amazon Blackwater, you would get these very red color profiles of the water there. And this methodology is really cool for vision scientists because it's really great to have a complete snapshot of the light that an animal's interacting with visually in its habitat. When we want to bring animals into the lab to try to test their visual systems, we often just put them in an arena with not too much thought about how much it replicates the natural light habitat. And now we're trying to use this data we've collected in their actual habitats to create new marine aquarium arenas to, when we're testing their visual behavior, we want them to be in as idealized a light environment as possible, matching their natural habitat. And beyond the applications for vision science, I think there's a lot of other cool applications for this technique. One would be in design. This is a ELF data set from our departmental office areas. And you can see that looks nothing like any of these natural scenes that I've shown you previously. I think it'd be really interesting to try to come up with design concepts to mimic these vertical light gradients of nature inside, to make a room feel like a forest or a desert or a coral reef. I think that's a really interesting direction to take this in. Also for agriculture, we can measure what a indoor animal facility looks like compared to a pasture where the animals are outdoors in the summer. And we could perhaps take some of the information from this and try to better illuminate their indoor wintertime stables in order to make it more closely and match their pasture habitat. It's also a way to look at human impacts. So you can get these images for light polluted areas, and you can see they're incredibly unnatural, this red mush that is very unusual compared to natural scenes. You can also look at climate change impacts with this, where when corals die or when different types of algae invade a marine ecosystem, the vertical light gradient changes quite a bit. And beyond that, I think there's a lot of just artistic merits to this type of system for looking at environmental light. Just the snapshots that make up these averaged images on their own often are quite beautiful. But you can also, I think, get a lot of visual appeal out of some of these averaged images. And they give a sort of impressionistic feel of a habitat, of a forest or whatever we want to think about. And you can also do it in a less random way where you're intending to try to create something with this system. For instance, this is a image of walking around a lake, shooting always at the lake, walking around a church here in Sweden, walking around a tree, and walking down a path and then averaging it all together. So I think there's some really interesting things you can do with this photographic technique. But with that, I think I'm out of time, and I'd like to acknowledge all of my collaborators and other researchers that helped with some of this work, and all the funders that helped fund this research. And thank you to the organizers of this conference. And if anyone has any questions, unlike this fan worm, I'd be happy to answer them. Thank you.
Yeah, Michael, thank you very much for this absolutely hilarious presentation. And is there some questions? Yes.
Hi, thank you. Thank you very much for the talk. So I wanted to ask about the Manta shrimp, as you were talking about the processing of the signal. Could it be that the color perception is not color perceptions per se, but it's only to have enough light stimuli coming in? So when we are comparing human vision with three cone types, you can see that the peaks are quite broad, but with the Manta shrimp, you can see that they are pretty close. Pretty steep, let's say. And it may be possible that that actually that's because they only try to get as much as light as possible to just short span of wavelengths.
Yeah, no, I definitely, that is definitely what they're doing in some extent. They're, by having these very, we call them narrow photoreceptive spectral peaks, they're sort of moving the processing out of the brain and putting the processing in the retina by having these very narrow chunks of color that they're analyzing with each photoreceptor. That allows them to not have to spend so much doing this ratiometric comparison of these broadly overlapping receptors that we do. And then that is one of our main hypotheses of why they built their color vision system like this. And it's a very different path to a color vision system. It's more like a color recognition. You might not even call it color vision without the ratiometric comparison of overlapping receptors. That can be debated a bit, but yeah, we do try to call it something different, color recognition or color barcode scanning or something like that.
Well, also the Manta shrimp are pretty colorful. Do you think that, or do you have any evidence for coevolution with the color vision and the complexity of colors on the surface of the Manta shrimps?
Yeah, that's definitely our number one hypothesis for what the color vision system is tuned to look at. And certainly the color would be co-evolving with the ability to detect all these gradations in color. They definitely communicate a lot of information to one another with the colors on them. Species that live in overlapping habitats with many other species of Manta shrimps are especially colorful. And they actually have a specific part of their anatomy. On the inside of their raptorial appendage, there's a colored spot. And each species will have a specific color on that spot if they're living in a habitat with lots of different species. So that's definitely a quick assessment they can do to tell which species of Manta shrimp they're looking at, whether it's their own or a different one. There in general seems to be a huge pressure for them to evolve colors that are very rich and very interesting patterns to them, and then also at the same time evolving the spectral filtering techniques in their eyes to be able to detect these gradations in color.
Thank you very much.
Is there some practical use to this special scanner?
Yeah, there's been quite a bit of work by colleagues that are interested in making camera systems that work more like this Manta Shrimp Eye. And there's actually been some really interesting work, especially on the polarization vision system. Some cameras that have been made that replicate how the polarization sensors in the Manta Shrimp's eyes work have really interesting applications. They can detect, for instance, cancerous cells within a image of a normal organ or tissue. just because of how polarized light is reflecting off the cancerous cells. There are also applications for environmental monitoring, for looking at from satellites or from other types of sensors, looking at changes in the color and the light quantity and quality coming off of different areas in the world, to see effects of like climate change or any other sorts of changes in nature. And also, there's some optical components inside the Manta Shrimp Eyes that are perform better than synthetic versions of these things that humans have designed. Again, in their polarization system, they have these quarter wave plate retarding structures that work much more efficiently across all wavelengths than some of the synthetic ones that humans have created. So there's a lot of possibilities for bioinspiration, looking at how evolution has built a really interesting color vision and polarization vision system in these animals.